From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 87, recorded on February 21st, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Great to be with you from LD Lab Studios in the snowy Wasatch Front, the Western Rocky Mountains. Winter's going strong over here.
A couple of weeks ago, did a This Week in Neuroscience with Jason, and he said they had 5 feet of snow or something like that.
Yeah, so the main ski resort here, it's north of 500 inches base at the moment, or total snowfall, I should say. I think their all-time record's like 840. I don't think we're going to get there. I hope actually we don't get there. That could mean some flooding and related stuff, but it's, as the kids say, it's nuking right now in terms of the snowfall. With a big storm. Yeah, that's right. With a big storm coming in here. Hey, Vincent, I have to say your voice sounds a little raspy. What's going on over there?
I have the radio voice. Yeah.
Yeah, there you go. That's right. You've been working.
Been talking so much that it's become raspy. No, as I told you, I had a little surgery on Friday, and I got a tube put in, and they probably scraped up a little bit of the pipe going down, right? And that's what that is. Because I haven't been talking all that much today, in fact. So it must be remnants of that. But I'm almost ready to go back to the incubator tomorrow, I think.
Oh, very nice. Yeah, sounds like a good, smooth recovery. Well, the good news is we have another voice to join the choir, so to speak.
Yeah, I'll let you two guys do most of the talking.
There you go. Yeah, so I'll bring in our guest right now, and then maybe we can go around the horn and say hi to the folks who've joined us on the live stream today. So I'm really excited to welcome my colleague and floor mate here in the Department of Human Genetics at the University of Utah. This is Nathan Clark, Associate Professor of Human Genetics. Welcome, Nathan.
Thanks so much, Nels. Thanks, Vincent, for having me.
Pleasure. It's cool that you're right across the hall from Nels. That's perfect.
It's a small world in science.
Yeah.
Yeah, I see the other side of the mountains over here.
Cool. Well, we're going to talk about one of your papers, which was sent to us, I think, by one of our listeners a couple of weeks ago. But before we do that, I want to thank everybody for coming today. We have 50 people here, and our moderators, we have Tom, we have Steph, we have Barb Mack, and we have Andrew. Andrew is from New Zealand, our newest moderator. Yeah, thank you all for joining us.
Good to have you all along for the ride. Thank you for all you do.
So let's see where people are. So Tom, one of our mods, is in Madison, Wisconsin today. I know that Nicola's in Italy, right, Nicola? Tom is in Madison, Wisconsin today. Gary is in Colorado. Lori is in Texas. Noir is in Santa Fe. Claire is in the UK. My alter ego is from Metro West, Massachusetts. Steph is from San Francisco.
Hi, Steph. Good to have you back.
Jan is from Tucson. Matt is from San Francisco. Pete is from London. Other Shade is from Pete. Amsterdam. And here's Andrew from New Zealand. ME is from Helsinki.
A little chilly up that way.
Barb Mack is from the UK. Look at this, Kim, another Finn here. I can't pronounce that. Can you pronounce that, Nels?
Well, I can't. Nathan, do you want to take a run at it? How's your Finnish language skills?
Jyväskylä. That's my try. We know a couple from Oulu, and they're wonderful people. And I don't know if that's anywhere near that spot.
Eastern Massachusetts, Buda, Texas.
All right.
Buda, I've been there. I've got my family's in Austin, so.
Berlin. That's cool. Dayton, Ohio. Yeah, Nicola's from Italy. Birmingham, UK, Connecticut, Boston, New Hampshire, and Vancouver. I am always amazed at the spread of people here. Another one, West Virginia. So this 60 people from all over the world essentially come to listen to us talk about science. What could be better than that, right?
I agree.
Welcome everyone.
Yeah, good to have
I
you
think
all.
This live stream is just so cool. Nels caught on quickly and realized that you could have all these people listening and asking questions at the same time, which is very cool. And not just locally, but all over the world. Milos is from Serbia. Welcome back.
Welcome.
Okay,
yeah,
so
let's do this. So, as I've said, I can't get enough of this. Sort of like opening a window into the world, and surfing some science together. And some of our Texas listeners will soon be circling back there. So, Nathan, why don't we start by just talking a little bit about you, where you're from, and a little bit on your science journey.
Awesome. Well, thanks. I guess my science journey started as a kid watching Nova and Connections and Cosmos on PBS, right, in a suburb of Dallas. Went down to Austin, Texas. Went to the UT Austin State Schools, a good school, a good price. And there, I majored in organic chemistry, believe it or not. But I guess I had a side major in trumpet. Basically, I was playing in all these kind of ethno-musical ensembles, salsa bands and Brazilian music and things like that. There's a ton of great music in Austin, and was tempted to jump ship from time to time. And a couple of my old bandmates went on to win a Grammy, but decisions were made. And so here I am, loving life though. Continued, organic chemistry was amazing. I was one of those rare people that enjoys it. I thought it was just the greatest thing in the world. But at the end of the day, I felt like too many of the questions were answered, and I wanted more of a challenge. So I went and traveled the world for a bit, taught science in a high school in Mexico, actually. Not New Mexico, in Monterrey, Mexico. Some of the best rock climbing in the world there. And after that wonderful experience, came back and applied to grad schools and was working this temporary job at UT Southwestern, just kind of making microbiology materials for the med students. And I met this brand new professor who worked in C. elegans, and he said, "Hey, you got to check out this brand new program in Seattle called Genome Sciences." And it wasn't even on my radar. Well, it turns out that was a fantastic meeting, because that's where I went on to do a PhD in this new field of genome sciences. I was in the very first class there there were 5 of us at first, and learned to kind of see the world through a massive scale, entire genomes, not genes, but what can we do to compare all these genomes to each other, and not worrying about how technology it's going to come along. What are the questions we can answer?
Yeah, this is a great department. So now on to, was it just the 20th anniversary that just happened for genome
That's
sciences?
Right.
Yeah. And so this was, in its origin story, this was a merger of the old Department of Genetics with the Department of Molecular Biotechnology. 20 years
Lee Hood's
ago.
department there. Yeah.
Mm-hmm. And Stan Fields, I think, was kind of one of the early sort of promoters of this. And I would say what massively successful 2 decades for exactly the reason you're describing, Nathan, that, to think in terms of genomes instead of genes, and what that opens up when you start to bring in the technology as sort of a 50-50 partner in the pursuit of science. Pretty exciting. Can I circle back for a second though? So how long was your gap between undergrad and grad school? How many years did you take teaching, et cetera?
So my intentions were to go down for 6 months and come back. Having played trumpet and salsa bands and stuff, it always bothered me. I didn't understand the words, but it also just, I wanted to know more about. I wanted to go see Latin America, right? So those 6 months very quickly turned into 4 years.
There we go. Wow.
But only 2 of which I was actually down teaching in Mexico. That also includes about half a year backpacking around with my wonderful girlfriend at the time, who is now my wife of 18 years, where we backpacked around Brazil, Bolivia, got a bit of Peru and Paraguay in there too. But yeah, just wandering and having a great time. Yeah.
Yeah, no,
And so one of the nice things, I mean, I encourage this in some people, I wasn't ready to go directly into grad school. I was coming out of, I just had things I wanted to do. And I knew once I got on that track with the way I envisioned becoming a scientist, I wasn't going to have time to do things like that. So I jumped and did it and did everything I wanted to. I rock climbed a lot and made friends from all over the world. Monterrey is this big business center in Mexico. So you get people from literally all over the world. Learned Spanish, traveled, saw all kinds of great things. And then when I came back, man, I was ready. I just knew, this is, after all that, I knew I had such clarity, and I knew PhD, hopefully have my own lab someday. I was 100% committed at that point. So that was a necessary thing for me. And I've seen this in other people as well, right? If you're feeling that, don't be afraid. Just make sure you line up your letters of recommendation before you leave so they don't forget who you are. How's that?
Yeah, pretty good. 4-year gap. That's right. Time flies when you're having fun.
Yeah.
So then
I vote for the gap too. I took a year off, worked in a lab for a year and decided that I wouldn't mind pushing small volumes of fluid around for the rest of my life. So
clear, colorless liquids. Yeah.
There you go. Exactly. Yep. So then you made it to Genome Sciences, and did you know which lab you were going to join right away, or how did your PhD unfold?
I left it open because remember, I was coming from organic chemistry. The only research I had done was making polymers for microlithography, making computer chips. Again, another career decision. I think if I'd continued down that road, I would already be retired with a couple homes around the world. But decisions were made, and I think I'm a happier person anyways. And so I think I was leaning very heavily towards joining the lab of Stan Fields. Wonderful guy, innovator. He created the yeast to hybrid, this kind of extremely clever way of finding protein-protein interactions just within yeast and all kinds of new saturation mutagenesis, things like this. But then I had a great visit with his lab, and then I did one of these rotations with this brand new professor who wasn't even there when I interviewed, named Willie Swanson. And he just blew my mind with how he could take protein sequences from a collection of different species and infer what are the important portions of that protein and when specific changes happen due to adaptive evolution that kind of pointed the way to historically over millions of years what's been going on. And so I caught the evolution bug, and so choices were made.
That's great. Yeah, fantastic. Yeah. And I think we'll still see some of your, the PhD training continues on as you're exercising science today, and even in the paper today, some of that training. But I guess so maybe a few minutes just on what you did for your PhD, and then that transition to the next step after that.
Yeah. So the Swanson Lab's really well known for studying reproductive proteins. So the proteins that mediate interactions between sperm and egg, from different individuals, and kind of how they change over time. And at first, what they discovered in allied labs was that these proteins change extremely rapidly, which was a big shock. People thought reproduction is so fundamental, it must be very, carefully guarded and conserved over time. These things aren't going to change. If you have a sperm and egg that aren't, interacting correctly, well, that's a huge hit to evolutionary fitness. So they should just never change. But the more and more people looked, those specific molecules crucial for the interaction and recognition of sperm and egg were the ones that were rapidly evolving. And so a lot of our work in that lab was showing that this is due to adaptive processes, most likely due to competition between males for a limited supply of eggs, but also due to females and males having different optima, different interests, different needs. And so the females kind of change their egg proteins to suit their needs, and then the males change, and that causes the females to have to change again. And it's kind of it's an arms race, just as the type you see in virology all the time, right? But it's going on within our species, between male and female gametes, right? So I jumped into that ballpark as a PhD student and kind of quickly expanded. I was curious to see people had done these studies in things like marine invertebrates, like abalone and sea urchins, and in Drosophila, the fruit fly. And I was curious to see whether this held in primates as well. It's like, is this going to hit close to home? So I started doing these kind of investigations into human and chimpanzee and other primates, and basically found the answer was yes. There's rapid evolution going on of these sperm and egg proteins and accessory proteins as well within our own home here. So we're really no different.
Yeah, and I love that sort of kind of flipping biology around, where now the essential processes can also be the ones where you see this rapid evolution. The whole era of model genetic systems, right, was sort of written on the idea that the most important stuff, the essential stuff, is where conservation is. Those are the things you'll find in abalones to humans, but this sort of opens up a whole new world to think about that essential things can be changing just as as fast as anything else in the genome. So this is on a personal note, Vincent. This is where I think Nathan and I first sort of probably collided scientifically. As Nathan was finishing up, I think I was just arriving, thinking about rapid evolution, kind of from that virologist angle or viewpoint, and how immune functions can evolve rapidly. That was when I was doing my postdoc in Harmeet Mullick's lab over at the Fred Hutch. And genome sciences isn't too far. It's like a 10-minute shuttle in the back of an uncomfortable bus to get over there and compare notes with the Swanson Lab. And I think there still are great conversations going on along those lines.
So did you know Nathan at that time, Nels?
Yeah. I don't know if you knew me, but I knew you. I was like very much excited about the work you were doing.
But say that in that last year, I definitely knew you, Nels. Yeah. And we, but at that last point, our first child had just been born, and I was trying to write a dissertation while not sleeping. And so we didn't have a lot of great chances to interact, but those came later.
That's right. So, and then plus you're writing that dissertation and going on to the next thing. And so and it was a pretty big leap, at least geographically, as you did your postdoc. So give us a little bit of that.
Absolutely. Yeah. I hopped over to upstate New York, right, which is a really cool place, Ithaca, New York, right? And so that's where you find Cornell. And I went to join the lab of Charles Aquadro, more frequently known as Chip Aquadro, who's kind of one of these pioneers in applying evolutionary and population genetic thought empirically, right, in the genetic age. So bringing in genetic tools to this. So that was a whole lot of fun. And what a cool community out there. It's just such a small town that you go out just wandering around town and you bump into professors and other grad students and stuff around. It's that kind of small college town experience. And there, I had all these kind of unformed questions about how to study fundamental properties of how proteins evolve. And, some of those went by the wayside because I started messing around with this idea that came when I was still in the Swanson Lab. And that was to study, how's the best way to say this, kind of the rates at which proteins evolve over time, over a collection of species, and looking for patterns in those rates over those species, and linking together proteins that have similar patterns. And it turns out this is a very nice way to infer proteins that perform jobs together in the cell. Just so not doing any experiments whatsoever, just looking at their sequences and picking out ones that have similar evolutionary patterns. That's basically it. And that's kind of what took off working during my time there working at Cornell.
Yeah. I used to say that computational stuff wasn't an experiment. I got yelled at a lot. So now I think it is a kind of experiment, isn't it? It's not a wet experiment, that's all.
True. Yeah. All in the computer chip.
Yeah, exactly.
In silico.
Yeah, so it requires controls and cleanups and quality control, just like any good experiment. Yeah.
Yeah, agreed. So then that you used that work and that concept of coevolution, or and we'll dig in on this as the paper from today. We'll say, but so tell us a little bit about that transition then to opening your own lab. And so I should say Nathan didn't open his lab at the University of Utah. That came later. So yeah, bring us along that part of the journey.
Yeah, sure. So those computational methods took off, and I applied for faculty jobs. I don't know how much we're going to go in the weeds here. This has to do with kids again. We had 2 more kids there in Ithaca. So postdoc salaries, I'm so glad they're going up now. And it would have been nice if they had gone up then, but they need to. Postdoc salaries here in the U.S. we were losing money quickly. Let's just say that. And so it was a do or die year. I needed a faculty job, so I applied to 71 jobs. And I landed one at the University of Pittsburgh in this fun department called computational and systems biology. And there, for any of you who are aspiring scientists and stuff, you always hear about this imposter syndrome thing. And I had it really bad. A lot of people did. I came in and I was just completely mortified at the idea that they had so much confidence to give me this space and a large chunk of change. But as I started to get papers published and things came together. And so there, the lab kind of grew out of these ideas of using evolutionary patterns to make inferences about either protein-protein interactions, but also about finding, we made an alteration to those methods to find associations between specific proteins or specific genes, and environmental pressures, right? So the very first one we looked at was within mammals. We had this collection of about 60 mammal genomes. And we looked at those and said, hey, a lot of these mammals have gone back into the ocean, full-time swimmers, basically. That must have required a lot of genetic change to occur. And I wonder how much of that genetic change was shared between all of these species. So we lined them up and asked that question because it had happened independently for cetaceans, what we know as dolphins and whales, and pinnipeds, what we know as seals and sea lions, and again for manatees and so on and so forth, all separately. So what changes did they share? And that turned out to be a really pretty cool method.
Agree. Yeah. Agree. And this is actually where TWiEVO sort of comes into the picture, I think. So, and correct my timing if I'm wrong here, but one of those, thinking about the marine mammals and sort of shared features, not just, and we'll talk about sort of shared patterns of accelerated evolution or relaxed constraint, and we'll kind of unpack that in a moment. But some of the first environmental thing you were looking at, or patterns you were looking at, were the shared losses of genes. And so on TWiEVO 34, episode 34, this is titled, "You Don't Know What You've Got Till It's Pon." This is my way over the top attempt to bring in a Joni Mitchell tune here, which I'll circle back to in a second. But so this is about ancient losses of peroxidase 1, or paraoxidase 1, I should say, in marine mammals, including manatees, the loss of this enzyme. And then you linked in a really cool, and kind of, a heart-wrenching connection to pesticides that continue to be put into the environment. And so I went back to what's the album, Vincent? Little Yellow Taxi from Joni Mitchell.
Big Yellow Taxi.
Big Yellow Taxi. Thank you. You don't know what you got till it's gone. And there's one lyric that I worked on so much that I feel like I need to bring it back for our listeners again right now. This is like in the second verse or something. Hey, farmer, farmer, put away the chlorpyrifos. I don't care about spots on my apples. Leave me the manatee and the walruses.
It's good.
Well, that's generous, but near rhyme with with one of the pesticides. So sorry, Nathan. Why don't you give us a little bit of
beautiful?
Yeah, right. So give us a little bit of that story told by the person.
The original lyric was, "Hey, farmer, farmer, put away that DDT now. Give me spots on my apples. Just leave me the birds and the bees."
Yeah.
Right.
Much more poetic, obviously.
Oh, she's great. She was just great. Yeah.
Yeah. Beautiful parallel. I didn't realize the depth of the connection inside the song as well. Yeah. I want to say with that feature on TWiEVO it's definitely a lab highlight. And so thank you all. Yeah. I mean, as Nels was saying, we went in looking for, gene loss. What are all these genes we have? We share a lot of them with all of our fellow mammals. Maybe that's something we should point out from the beginning. Depending on which pair of mammals you're comparing, we share, somewhere around 80-90% of the genes that we have, right? But there's certain conditions that lead to those genes just blinking out. They're no longer needed anymore, or have become actually a burden, so they can be damaged and shut off, right? And this happened. We went into marine mammals, and we expected to see a lot of smelling and tasting genes, because it turns out they all lose those senses, believe it or not. And sure enough, we did. But right on the top of our list was paraoxidase 1, which made no sense at all. It's this bloodstream enzyme, and it has this serendipitous effect that it protects us from our own pesticides in the organophosphate clade. They're all neurotoxins, but because of this enzyme, we can tolerate a bit of them because we can break them down. But the insects do not have PON1, so they're very susceptible and they drop dead pretty quickly. So what we were horrified to find out is that basically all these whales, dolphins, lots of the seals, otters, beavers, and manatees are all as susceptible to the pesticide as insects are, because they have lost the gene. We're still trying to figure out why they lost the gene, but that's in this context beside the point. And we were finding that there were agricultural fields in Florida, was the case study we looked at, that were directly abutting freshwater canal systems that the manatees are known to inhabit, especially in the wintertime as they come in. So you could imagine the runoffs from chlorpyrifos being applied running directly into these manatees. It may not cause them to turn upside down immediately, but it could at least be contributing to lethargy, which might be leading to them getting hit by boats and things more frequently. So this is something to be aware of for sure.
Why do other species have the enzyme? Do we understand that?
Yeah, biochemists have done some fantastic work to show it's likely there to to get rid of oxidative damage to lipids in our bloodstream. So the enzyme kind of hitchhikes around on HDL particles, high density lipoproteins in our bloodstream. And anytime oxygen comes in and reacts with one of our fatty acids there, it causes those lipids to become sticky, and they form plaques, which leads to heart disease, heart attacks, and stroke and related things. So what these do is PON1 goes in there and makes sure those sticky lipids don't build up. It breaks them down to the smaller products that get shuttled out. So yeah, if you knock that out, if you take out that gene in a mouse, for example, it builds up large kind of dangerous amounts of these. And in humans, there are two different major forms of PON1, and one of those alleles, as we call them, is associated with a higher risk of developing atherosclerosis and heart disease.
Yeah, really interesting story. And so, and to kind of close the circle on, so Nathan's opened his lab at the University of Pittsburgh, but then here at Utah, we're starting to run a big hiring initiative in evolutionary genetics. And imagine our joy when we saw Nathan Clark's application in the pile. And so in the end, we ended up hiring 5 new faculty to join. Talia Krasov, who actually has been on TWiEVO. I think we met her at the annual American Society for Microbiology meeting a couple of years back. I don't have the TWiEVO number right off my fingertips here. A fellow called Dean Castillo, another guy, Michael Werner, and then and they're all faculty in biology here at Utah. In genetics, Nathan and then Ellen Leffler, who's my next door neighbor here, both joined, with Nathan coming in kind of that mid-career zone, having already started his lab at Pittsburgh and then joining us here at Human Genetics. Then the pandemic happened and the whole world kind of got scrambled. But really, really fun to not just admire. So kind of going from that 1 year of interacting a while ago in Washington to now being colleagues on the same floor. This has been sort of shot in the arm, not just for my lab, but for the whole sort of academic energy here around the growing community of evolutionary genetics at Utah. So that was huge.
Very kind, Nels. It's great to be here too. It's good to.
Nathan, what was the impetus for moving?
Oh, wow. How much of the inner workings of medical school do you want to hear?
I spent my life at one, so I understand.
How much do the listeners want to hear? That's it. Let me try to get the department I was in, computational systems biology, is really great people. What got me was the opportunities here, first of all, Utah, to be with that fantastic team. Also, a bit of a funny thing, the chancellor and kind of dean of the School of Medicine back there in Pittsburgh had always loved evolution, and he had been talking to me and Vaughn Cooper about creating a department for evolutionary biology and medicine, basically. And so 5 of us got together, really great group, Dennis Koska and Eric Wright and Rixandra Carvunis and Vaughn and I, and we wrote a white paper and we delivered it to him, said, we're ready. And well, it turns out he couldn't do it because they were trying to force him out. So they had taken away the dean's money. And so kind of the wind fell out of the sails there. And then the dean split our department to 2 different buildings, because he, as his retirement project, he wanted to start a new neuro institute. So I saw the way the winds were blowing, and caught one west. Right.
It's so unfortunate that the academic politics always has to wedge its way in there into the science.
Yeah,
it is a bit odd. Yeah, but onwards and upwards, and things are looking good. Yeah.
Yep, agreed. So small world. So then, bringing us up to today's paper, I get a text message from Vincent, late at night. That's how we communicate here as TWiEVO co-hosts, is we see something cool comes across our radar and we say, hey, we got to do this for the live stream, back and forth, et cetera. And so someone, I think, reached out and put this paper from Nathan's lab on our radar. And I thought, not only let's do it, but what a great opportunity to grab Nathan from across the hallway, get a microphone in front of you to give us the whole backstory here. And to just say, Nathan coming to Utah and being across the hall, maybe this is all, there's the medical school politics, but maybe this was just all a ploy to become a guest on TWiEVO is my sort of secret. I'm joking here, but anyway, so let's talk about this paper. So complementary evolution of coding and non-coding sequence underlies mammalian hairlessness, and as a facial hair wearer, this one strikes a little bit close to
shocking.
Home. I hadn't realized this was even a possibility, that you could lose all your hair as a mammal over here. But anyway, to jokes aside, and on that personal level, maybe tell us a little bit on the backstory of this paper and how the project came together, and give us the intro here.
Yeah, happy to. Yeah, so with these methods that we had kind of honed on marine mammals and other groups, we're always looking for new traits to study, right? And the wonderful PhD student, now Dr. Amanda Kowalczyk, approached me and she said, hey, there's this great trait that another professor we were working with named Maria Chiquina had mentioned to me. We could just very simply look at these species, and a lot of them have lost their body hair, almost all of it, right? And I was like, well, at first I wasn't that enthusiastic. I was thinking in terms of, funding a lab. There's no National Hair Institute out there to, it's not as important of a problem as cancer. So I was like, but you know what? Amanda was with her fantastic research hands. I said, you go for it. Let's find what we can do, because it is interesting. I still have curiosity deep within inside me. So Amanda started at this, and she just very quickly, it started working. As you would expect, some of the top genes to come out being kind of lost in, and well, first of all, what are these hairless species? If you think about them, right? We were always told from our first science class that hair is kind of one of the defining characteristics of mammals. And I think it still is, because even all these, what we were labeling as hairless species still have some. You look at an elephant, in juvenile forms, they'll often have kind of rough hair on top. Even adults will have kind of sensory hairs on there. You don't see them, but they're there. Manatees, have lost their hair. A lot of kind of streamlining underwater. Naked mole rats, armadillos. Some of them actually have furry bellies, but they've lost most of it. We classified ourselves as humans as relatively hairless, hair reduced. Right? So that's kind of the, rhinoceros is this kind of.
Well, Nels is not hair
range.
reduced. He's got lots of hair.
It's getting worse almost by
it was tempting not to put humans on there. We had to. It's funny when you start trying to classify things on such a simple split, how complicated it could be. We didn't even know what to do with pigs, because we're used to farm pigs, but they're wild relatives. These boars, they've got hair. And so where were we going to classify these guys? We
what
had to
about
think deeply about
dolphins? They don't have any hair at all, do they?
Embryonically, yes. They have been shown to have some, and then it disappears before they're born. So, but yes, they were a no-brainer to go in the no hair category.
Well, and this sounds exactly like a Clark Lab. Just the setup is just right in the wheelhouse of the Clark Lab, which is multiple independent changes, and then using evolutionary sort of signals to go at this. So how can evolutionary analysis point to, in this case, the genetic basis of reduced hair quantity?
Absolutely. Well, so here's some of the nuts and bolts. So buckle up. We have ways of looking at the rates at which at which genes change, and we can kind of measure and determine what selection pressure has been acting on them in the recent times, and to what degree. And those selection pressures are conservation, which slows down the rate of change, as well as two forces that can lead to acceleration. One is if a gene becomes unimportant, its rate of change will accelerate, or at least less important. Or in cases of adaptive evolution, that will also accelerate the rate of change. And so what the idea in these cases, if we see that if any given gene changes its selective pressure the same way in the hairless species, but doesn't change in the hairy species, then that's the kind of signal that our statistical test will pick up. And in this case, we expected this to be mostly loss of function, right? If you're not producing as much hair or hair in certain places, genes involved in that will tend to be more rapidly evolving because they're being released from constraint. They accelerate from our point of view. And the reason, as Nels mentioned, it's important to have multiple cases of this is that, unfortunately, if you look at any one species that has lost its hair by itself, there are going to be millions of changes in there. And it's a classic needle in a haystack problem. You're never going to find exactly which ones were specific to hair loss. The genome is a very big place. But if you have multiple examples of this repeatedly, you get kind of what's akin to experimental replicates. And you can look and say, what are the genes that are recurrently being released from constraint? And we found those to be, first of all, genes like keratins and things that are known to be important in the formation of hair shafts and things like this. So those kind of served as what we call positive controls that let us know we were on the right path.
Mm-hmm. So, but you also had this tool already in hand. And so like your point from before, Vincent, is really important here, which is sort of bringing that notion of kind of an experimentalist framework or viewpoint to computational analysis. And that's what you just connected it with, Nathan, with your idea of these natural experiments happening. But then, if Vincent and I were going to try to search for those needles in a haystack, we'd be lost. But you've got a tool that does this. So tell us about that, RER Converge, and how you can deploy this to actually tease out those natural experiments.
Sure. Yeah. As we like to say, nature has performed countless experiments. We just have to sit back and interpret them. So RER Converge is an R package that our lab has written jointly with Maria Cecchina's lab back in Pittsburgh. What it does is it takes you begin with a set of of genetic elements. Those could be genes, but they could also now be regulatory elements that are found between genes. And you collect them from a large number of species and make gene trees from those. And those are what serves as input to our package there that you can run in the R statistical environment. Once you have big collections of these different genetic elements from the same collection of species, it does a lot of kind of nice normalizations in linear algebra. And then you get out these things that we call relative evolutionary rates, which is a rate of change for any particular element in any particular species, which is an enormous matrix of millions of entries. But then you can kind of do what computers do well and comb through all of those different genetic elements, looking for the very best that match that evolutionary pattern you were expecting to see based on your trait, in this case, hairless species, right? But you could take those same values and polarize them on any other trait you want to look at. We've looked at high altitude species or species with poor eyesight, and those have all yielded really kind of cool insights into new genes and pathways involved in those processes. So essentially learning from the loss of something, you can learn what's important for it.
Yeah, no, it's really cool. And so millions of comparisons set up for computational analysis, but how many comparisons do you need to make here? How many genomes do you need, or what's available for you to do this? What's the substrate for this analysis?
Yeah, so it's becoming easier and easier. When we were first doing these types of things, we were excited to have 15 species. We were doing some in the fruit fly based on 8 species back in the day, but now we're in the day of where genomes have become very cheap and routine to produce. So now we have, within mammals, for example, we have a collection of something like 300 species that we can operate on. We have, within yeasts, we have 350 species we can operate on. In Drosophila, fruit flies, you have more than 100. I'm sure if we started, we need more hands on deck here, because we could start getting more into microbial and pathogen worlds as well, because you've had hundreds of genomes for much longer, right? So you can do it with as few as 10, but we've been able to show that you have more and more power now with hundreds.
Yeah, no, and I think this is an important point to underline, Vincent, is sort of this moment in evolutionary genetics, evolution, where it really is a new playing field in terms of the quality of the genomes that are coming out of the diversity of life. And we're still just scratching the surface, but very soon, very quickly, you can fill sort of like an entire compute cluster with the amount of information that you can crunch through and unlock all of this biology that just wasn't possible. Would you say even like a few years ago, this wasn't possible?
Well, I like to make the example of my postdoc work. I sequenced a virus genome, one of the first. It took me a year, and I wasn't going to do any more than that, right? So you would get one genome, and you worked on it for 10 years, and that was it. And then maybe 10 years ago, it changed, right? When 454 and then other high-throughput methods emerged, that really changed the field to the point where now you could do hundreds and then thousands of viral genomes, and you could look at evolution and see what changes are associated with that, and then you move on to other species. So it is a total change in the way that we look, and that's why in part we need computational power, which came into the field just as the genome sequences were increasing in number, and that's what we needed.
Yeah, agree.
Absolutely.
Would you say?
Yeah, let alone we had to rewrite all of our functions to work to be able to function with these large numbers that we weren't anticipating anytime soon. We went from something that was pretty much infeasible, it would have taken maybe a year to run, to now we're down to within 20 minutes. So it's amazing when you get real computer scientists involved, how they can speed things up a lot.
No, it's the same with crystallography, right? X-ray crystallography. You couldn't do very large proteins, right? You did myoglobin and hemoglobin, but when it came to viruses, you didn't have the computing power to do it, and that was the game changer. Yes. Same thing with cryo-EM. The resolution increases with computational approaches. So despite most of us who were trained on wet experiments, feeling these intruders coming in, it's necessary. You can't move forward without it.
Yeah, it's important to keep nurturing labs that kind of bridge those two areas, because it fosters kind of collaboration and communication between groups. We're needing to become more and more specialized.
There has to be a collaboration, though, because I hear now everyone is saying, "Oh, AI is going to revolutionize biology," but you need to put data into AI, and if you don't, they're not going to guess things. They have to have a grounding for it, and to get those data, you still have to do wet experiments. So you have to work together. Yep.
Yeah, agreed. Yep. I'm curious, Nathan, how much of a bottleneck would you say that good genome annotation is at the moment to make sort of robust comparisons, or is that even advanced to the point where you're feeling? Yeah,
it's huge. It's a big challenge. Yeah. Mm-hmm. Now producing the raw data is no problem. Even assembling has become, a much more tractable problem. Putting all those letters together into one massive genome, determining what is a gene and where they are and what is, all that is annotation, as you're saying, is huge. That's a major challenge.
Yeah,
for sure.
Yeah.
So Nathan, can you give us a sense
for us?
Can you give us a sense for how many protein coding and non-coding regions you found would track with hair loss?
So it's tough to say that, because it depends on where you draw the line of confidence. But I would say where I draw my confidence line, it's certainly around 100 are pretty confidently predicted. And as I was saying, the good thing about those is a lot of them were proteins that we would have expected based on work on hair, but there's meshed into there all these other genes that we don't really have a good grasp of what they're doing, and that's one of the reasons of doing this, right? So we automatically, the idea is these should be tested for their role in hair follicle formation or hair shaft growth, all these different steps. And we even found a couple of microRNAs that were implicated. Yeah.
So this wouldn't be something you would do in your lab. You're hoping other people would do this, or am I wrong?
Correct. We don't have a hairy mammal model in our lab. We don't have any mice or anything. We have zebrafish, and so we actually did take a couple of the regions that we believe are regulatory regions, and we plopped them into a zebrafish to see if they would perhaps be driving expression in scale progenitors or something kind of analogous to hair. And that's still ongoing. But Amanda, Dr. Kowalczyk, has reached out to a couple of labs that do study hair follicles and stuff in mice, and so we're trying to push work forward there. But my concerns have come to fruition from the beginning that not a lot of labs work on hair in academia because it's not a huge health issue, definitely self-esteem issue, right? But perhaps maybe we're looking in the wrong places. Maybe we need to start approaching hair loss companies that would love to work on some of these genes.
Well, and that is a major industry, obviously. I mean, the Rogaine industrial complex is not. That's for real.
I was going to say we have a professor at Columbia, Angela Cristiano. You may have heard of her. She studies hair because she had a hair problem of her own, and she got at the genetic basis of that, and that is big bucks. People don't like to lose their hair, so
no, they don't. No.
Both men and women, and you might find more grant support for that than why a dolphin lost its hair, right?
Well, that's the trick, right? Is so using these comparisons to unlock the secrets, these natural experiments to unlock the secrets of what might be going on with gain or loss in this case in our own species. I was going to say I'm happy to volunteer as a hairy mammal at any point, Nathan, if I could but okay. For experimental scenarios. But so one of the keys here, though, I would say from an evolutionary standpoint, is sort of the evolutionary of hair in mammals. So I guess is the idea that the ancestor of all mammals, was this like a mouse-like critter that was very hairy, or I guess you start it there, and then the hope is as the lineage became diverse of mammals, and then that allows time for these independent losses to occur so that you can make your comparisons. But where did this all start from?
Yeah, well, this is going to involve some speculation here, but certainly the ancestor of all modern mammals had hair, right? It's clear because the way it's formed and all is common between all of us. How much hair it had, it's actually hard to say. It was a warm environment back then, so it might have resembled more kind of a lot of tropical mammals that rhinoceroses, elephants, but naked mole rats that didn't have any, although it's likely to have had a few. Even tunneling mammals still have some now. Since then, you've seen it specialize in many different ways. It's protection from UV, it's protection from other damage, and it's an environment, or perhaps even a bit of armor in a way, right? Signaling, you see, coloration on that hair becomes important for sexual selection in species. It's a pretty important mammalian trait, right?
So can you speculate on the sequence of events for, let's say, take a dolphin, right? So I presume the ancestor goes into the oceans. He's got hair, right? And that is accompanied by gradual loss because it provides a fitness cost to have hair in the ocean.
Yeah, that's the idea, right? There were these intermediate forms that were not free range ocean dwellers, but they were more kind of coastal, perhaps a bit more like an alligator is now, kind of existing in both realms. And a nice intermediate, even a little bit further committed to the aquatic lifestyle, is look at things like seals and sea lions. Some of them still have full hair. It's just slick, very close to their bodies, but some of them have lost it completely. So it's kind of tricky and I want to say, of course, they lost it for streamlining, but there are counterexamples, as is always true in biology. But perhaps cetaceans ended up losing it in their ancestor, like a walrus, for example, and just kind of kept it that way. They found other ways to insulate with blubber and things like that. So.
Yeah, and that's, I think, a nice thing about this approach is you sort of sidestep kind of evolutionary storytelling to just then ask at the genetic level or through the genome-wide scan, okay, well, what are the changes, shared changes in this case, to get those needles, like the multiple needles, I guess, showing up in the haystack to be able to identify what might be important here. And so it seems like the obvious cases that you've already been hinting at are some of the genes that have been already linked to functions for hair follicle development, et cetera, that came out of your analysis. But then you also kind of, took that sort of daring approach to also go after the non-coding regions. And so maybe walk us through that a little bit, or what motivated that idea and how did you do that?
Absolutely. Well, the motivation is we know so much less about regulatory regions than we do protein coding genes, right? There's roughly something 20,000-ish protein coding genes we can study in mammals, but there are hundreds of thousands, if not millions, of regulatory elements in the genome, and we can only really ascribe higher level functions to a very small number of those. So that's the frontier. So of course we'd want to get into that. For this study, we narrowed it down to a set of about 350,000 potential regulatory elements. We identified them by them being kind of conserved elements that exist, conserved in the sense that you can find them across a good cut of mammals, right? And we studied those. We don't know that they're all regulatory elements, but most of them probably are. Once we polarized those, we found great. A lot of them were significantly clustered near genes that were known to be important in hair follicle formation. And then it had some surprises as well. Deep into there was this, we asked this kind of question of where the high scoring regulatory elements fell, and there was this very strong cluster of them, very, very prominent. And so we zoomed into the genome to take a look on a genome browser, and there they were, all clustered, and there was no gene. It's just all, well, what are they regulating? And we kept zooming out, and there's still no gene. We zoomed out even further. It's like, what in the world is going? And there was this protein there, some CAM kinase protein, which is, of course, very important in nervous system function. And we're like, this doesn't make any sense. It's not even expressed in the right tissues. So we zoomed back in a little bit, and we saw this funny little thing that had been some evidence for transcription. And then we looked more deeply at it, and sure enough, it was a microRNA gene right in the middle of all those regulatory elements. We dug up some data, and turns out that microRNA gene is being expressed in skin and hair follicles. So perfect little story there. And we were able to identify 3 other microRNAs in the exact same way that are now strongly implicated in probably regulation of when and where hair follicles form. So those are the ones I'd be most keen to following up with our new Rogaine partners, for example.
Yeah, this is really cool. So this is sort of this genome hunting with not, so that microRNA hasn't been implicated in sort of hair development or regulation, and yet that sort of your analysis nominates it to be so, with just enough information sort of through functional genomics, I would say, to know, okay, this is expressed not in the nervous system, but in the skin or the follicles. And so then you can kind of prioritize the genes from there. And then I guess that's is that sort of the product you would say of this work, is to sort of nominate a number of non-coding or microRNA regions, as well as some genes that, similarly might sort of have some of the flavoring of this is involved potentially in hair biology, but haven't yet been really studied yet. So that's the kind of invitation for the next steps.
Yeah, precisely. That's probably the main product coming out of the paper, right? Is that now you have all these regions that we're fairly confident are participating in hair function. Right? Mm-hmm. It can also tell us perhaps a bit about the timing of loss of hair, but we didn't dig so much into that yet.
Gotcha. And you mentioned already, Nathan, so the notion of sort of accelerated evolution, and that it could be in sort of, that could be in different flavors. So relaxed constraint, meaning as mutations accumulate, they're not sort of selected or filtered out of a population, because you can kind of get away with a lot of different stuff there. The other sort of side of the coin, I guess, would be positive selection, which is that these changes bring some sort of benefit or advantage somehow. And so did you look at that or try to distinguish what might be going on there?
Yeah, that's a great way of stating it, Nels. So yeah, we were it's of course important to us to figure out if these were kind of drifting loss of function genes, or if they were adaptively evolving. And there are 2 ways we went about that. One, there are these kind of sophisticated models you can use to infer the past action of positive selection, of adaptive evolution. And we ran those models on there, and it didn't pull them up. Very few genes scored highly in that, which corroborated our intuition coming into this, that this should mostly be relaxation of constraint or loss of constraint. And in other ways, simply we kind of went through them and looked for things like early stop codons and frame shifts, things that should render a protein non-functional. And those were definitely more enriched in these genes than over the background, quite a bit more. So all signs were pointing to, in this hair case, being loss of function.
Yeah, it makes sense. Yeah. There's one other signal that you pointed out that I thought was really fascinating. I wanted to bring this up too, which is, it actually has nothing to do with sort of the phenotypes, the potential hair-related phenotypes, but it's the idea of the sort of the patterns of accelerated evolution, and how there is this, so if you're looking in the the genes that were implicated in hair loss, you might see this pattern of accelerated evolution. But then for the regulatory elements that you might propose nearby, those are pretty quiet, or you weren't getting signals there. And then the reverse is true. So if you have all of this action, relaxed constraint in the non-coding regions, then the genes seem like they're kind of, there's not a lot of change there. It's not accelerated. It's just evolution as usual, I guess. What do you make of that? Or why is that sort of an important signal to tease apart?
Yeah, that was definitely one of the cool upshots that Amanda picked out, Dr. Kowalczyk, that there was this kind of shift between the two. What we make out of it is that regulatory regions are much more important for kind of the origin and maintenance of the base of the hair follicle, right? And kind of keeping tight controls on that. And interestingly, in those types of, I guess, cell fate or kind of developmental decisions, a lot of the protein coding genes themselves get reused in other processes completely unrelated to hair in our body. This is well known in developmental biology, for example. So what you see is the proteins don't show that pattern in this kind of cell fate decision in the hair follicle, but the regulatory regions do, because you don't want to mess up those proteins for the other functions it performs. Whereas on the flip side of the coin, you see the proteins being degraded in the structural types of proteins that form things like the growing hair shaft, right? And those don't require as much fine control and gene regulation, so we didn't see signatures there.
Yeah, it's really interesting because there's this, we've talked about this, Vincent, there's this sort of long running, I don't know, maybe somewhat drummed up controversy over whether evolution, sort of the mechanisms of evolution, play out mostly in genes. So you make mutations within a coding region and then have these sort of, or if it's in regulatory regions that just can, like, which can you do, or which sort of dominates. But I really like this view, Nathan, because it feels like it's a little more, you've kind of contextualized it with the biology here in a good way. And it's not one or the other, it's sort of both, depending on how the biology is working, and that evolution will sort of act on that when it makes sense, in a sense. Yeah.
So absolutely, evolution is not being choosy.
Yeah, that's right.
We did a TWiEVO some time ago about birds, remember, Nels? And I'm wondering if Nathan's approach can be applied to loss of flight. Right?
Sure, could. Yeah. We have not done that, but a colleague actually, Tim Sackton, his group over at Harvard did that with some kind of related methods that we see as complementary to ours. Yeah,
has that happened enough times? So it's that convergence, that independent losses. Is that true for flight? I just don't remember.
Yeah, they're you got me cornered here. I'm not a bird expert, but yeah, from what I remember from that paper, yes, it has occurred multiple times. Yeah, and if you look at flightless birds like ostriches and kiwis and things like that, they're in different groups. But I'm just going to leave it at that.
I think it was
fair enough.
I don't want to misstate.
Flight was developed 3 times independently, but lost many more times. Right.
That's right. Yeah, that rings a bell. Yep. So Nathan, but what are some of the topics that you're using this tool RAR Converge in addition to the hairlessness, or the repeated loss of hair in mammals? What are some other examples that are like sort of these conversion events? I think you hinted at it before, but what are you up to these days?
We continue working on aquaticness in one way. We're also, a good portion of the lab works on vision. There are multiple cases where vision has been lost in subterranean mammals and bats and things that have poor eyesight, like shrews and armadillos. We pick on armadillos a lot, don't we, in our studies? I guess they keep coming up in these lost categories. They've lost so much. But we're also starting to look at other kind of gain of function traits. There's a wonderful study going on by a postdoc in the lab, Dr. Emily Copania, and she's studying kind of the development of retinal specializations. We have this ability to see and focus on very, in our center field of view. Well, that's not actually the norm. So we're looking at species that have repeatedly evolved that kind of central focus in their retina. We're also studying, through Dr. Ali Graham, we're also studying high altitude mammals. I have my sights set on mammals that have lost their teeth. There are a lot of different kind of toothless mammal lineages. I'll leave it at that. So those are some of the ones we look at.
How about land speed?
Land speed could be interesting.
Yeah, that'd be fun.
Yeah, muscle types. We've also studied longevity, so lifespan and body size kind of together in a study. That was really pretty illuminating. The upshot of that one was cancer prevention is very important if you're going to live a long time. So you want to highly protect and conserve your cancer prevention pathways, like cell cycle checkpoints and things like this.
We did a TWiEVO on that, didn't we, Nels?
Yeah, that's right. lots of fun ideas there. Peto's paradox is one of the
Absolutely.
ways. Yeah. So how do the elephants, the animals with long lifespans or a lot of cells, if you're a massive elephant, how do you get away with that without enhanced cancer rates of cancer? And if anything, it seems like they're lower. And so a number of labs trying to figure out potentially the genetic basis there. There was another one, Vincent, that sort of a local story that Nathan got involved in, which is one of our science buddies, James Gagnon. He just did a study on zebrafish, and the moms provide sunscreen to their offspring, maternal effect, a clear sunscreen. And so this has been lost multiple times in fish that have moved away from UV exposure, and so it's sort of the perfect. I can remember. Having coffee one morning with Jamie and him saying, what's the evolution of this? And I said, I've got the right guy for you. That was an easy one to say. Talk to Nathan, and the tools just lined up perfectly to see some similar signals, right? Or those patterns sort of emerging, the loss of this UV protection correlating with these independent life events, which was moving away from UV light as different fish lineages have done.
I have some questions here from our audience. If you would like
let's
to
do
take
it.
them here. Let's go all the way back to the beginning. Lori wants to know, this is with your manatees and so forth. Did you look at water mammals in other hemispheres, Asia, South America, and Australia?
Oh, for sure. Yeah, we had, within the aquatic mammal list there, we had otters from South America. We had Antarctic pinnipeds, leopard seals and wet-tailed seals, and we had dugongs, which the cousin of manatees down in Southeast Asia and Australia. Certainly we had a lot of breadth there that was provided to us by the genome sequencing communities.
Winfield wants to know, is there any research on how the hair-related genes relate to similar genes in Neanderthals, Denisovans, or Homo naledi?
That's such a great question. Yeah, we have not approached that yet, but we would love to do this. Looking at the relative timing of kind of when these things were relaxed, released from constraint and such, that could provide some nice clues. Yeah.
Yeah, that's a great question.
We've got methods we could start doing that, but we need to get to work.
Are scales, feathers, and hair related evolutionarily?
Okay, this is a bit controversial, and I'm not going to get too into this. I think they are. And other people do too, and they've published papers supporting that. However, there are counter arguments here that I won't have time to, I didn't prepare to get into right now.
Right.
I think they must have some analogous kind of gene regulation going on. That's what we would be looking for.
Mm-hmm. Mm-hmm.
But yeah, it shows that science is not, we don't know all the answers yet, guys.
Sure,
and
sure.
and it's, so yeah, encourage more people to get into that.
So Lee says maybe grant support for hair loss doing chemotherapy as well, right? That would be a way to do that.
Yeah. The follicles, the cells that turn over the fastest, which then are more vulnerable to chemotherapy. And if there's some clues that, and some new ones, like some of the genes that Nathan and Amanda are nominating here, then that could sort of unlock some of the targets or secrets to tackling questions like that. That's a really good point. Yeah.
Yeah.
So Nicole, a bit off topic. I'm a hardware nerd. Do you run all this analysis on a normal PC, or do you need something more powerful?
Yeah, we have, like most universities, we have some high performance computing clusters that we do a lot of this on. But actually, with the new speed ups that I mentioned earlier, we're now able to do these again on a laptop. If we want to do lots of statistical rigor, however, we can still put them up on the cluster, which are running kind of Linux type machines.
Barb Mack says hair loss during hypothyroidism is a big problem also. Another thing to look into.
Yeah. Thank you for that note. Absolutely. Yeah.
There's another one here. My question concerns the TOMP-rank gene, FGF11. Here we go. Not only is this gene implicated in hairlessness, it's also associated with tooth development in mice and dentition in Mexican hairless dogs. And so part 3, what selection pressures might account for such convergent evolution in mice and dogs? Does it matter that the morphological development of a tooth and a hair shaft are similar?
Wow. Yeah, so release from constraint, the genes, like we're mentioning, we share these genes, and it's not going to matter. Evolution doesn't really care which ones it goes through. It's does morphological development of a tooth and hair shaft, are they similar? I'm not prepared. We haven't done our toothless study yet. I wish I'd prepared for that. I don't know.
Well, and remind us, Nathan. So FGF11 was one of the we didn't kind of drill in or go into the weeds on that one. I guess no pun intended on the dentist drill situation there.
Right.
But so remind us. So FGF11, fibroblast growth factor 11, is this one of the genes that has sort of been, or it makes sense from where it's expressed and how it might be acting, but hasn't been directly linked until now to hair follicle development, or what was known about it, I guess, or why did you sort of really lift up that gene FGF11 from your analysis?
We lifted it up because it like you're mentioning, there already were studies showing it's some kind of master regulator in certain developmental processes. And as you're pointing out, it seems like it could very much be related to, unforeseen functions, right, that we're looking at. It could also be, and we should look more deeply at this, involved in kind of this regulatory evolution paradox that we're looking at, where the protein coding gene is important in other functions. Mm-hmm. So, is it changes through regulatory sequences that allow this change of function that we're seeing now without disturbing its other important functions, if that makes sense?
Yeah. So, I like that idea. And Matt, I think it's a great question too. It gets at certainly a theme and concept in evolution, which is pleiotropy. So, if something works, it can get caught up in sort of overlapping functions. In this case, both in tooth development potentially as well as hair development, and just sort of by contingencies or chance. Yeah.
And I should say that this FGF11 is kind of, I must admit, it's kind of haunted us a little bit because it is so important in other functions. So why would it be accelerating specifically just because of hair, right? And it could be that there is a certain portion of the FGF11 gene that is more specialized in control of hair function and growth. And that could be the part that has accelerated, whereas the rest of the protein has remained under normal functional constraint for those other functions. So that was definitely something we were discussing, but the final say on that gene is that we don't know.
Yeah, more work to do.
Step one.
Very cool. Yeah.
All right, Kang wants to know, is the need for hair different for aquatic and amphibious lifestyles?
Great question. I guess you have to ask yourself what you're using it for, like a fully aquatic versus semi-aquatic, right? And like I mentioned, some semi-aquatics have lost their body hair, but some have completely maintained it. If you look at amphibious creatures like otters and beavers are actually fantastic divers. They've maintained full coats of hair, perhaps because they're in cold environments. It can provide that needed insulation when they come out of the water, right? It's not doing much in the water. Yeah.
All right. And I want to remind everyone, we have about 100 people here. So if you haven't already clicked the thumbs up, please do so. We have 51 likes. We'd love to get as close to 100 as possible. Helps more people find us.
Yeah. And great questions. Really fun conversation here. I think
so. Something fun that kept coming up, if I may, is why, when people ask me about this paper, they all want to know, why is it that we humans have reduced our body covering of hair? Right?
Okay, yeah.
Mm-hmm. And we didn't find that through this paper, but one of the main schools of thought on that is that sweating is extremely important. Mm-hmm. That's kind of an ability we've taken to an extreme. It's definitely, we are the outlier there in terms of species to be able to cool ourselves in that way. So removing the hair kind of allowed that air circulation better, and so we could kind of outlast other species endurance-wise under the hot sun. So
interesting. Instead of having to do it through our tongues by panting, right?
That's right.
absolutely. Yeah, that's why you have these great stories of people chasing down game, just
right.
patiently tracking it until that antelope or whatever is just overheated and just walk up to it.
Yeah, apparently this is a new idea that humans could outrun, and maybe this is part of it,
Well, it does illustrate too, just like kind of the complexity of sort of the environment, the interactions that all come into play that get you to where you are, whether it's the climate or the predators and the prey, the heat, all of the above. Yeah. I'll just confess that, if I'm on my current hair growth rate, I'm just going to have to start panting again pretty soon because there's not a lot of open real estate left for sweating. Okay. Sorry. So, Nathan, we don't want to keep you here. If you need to, I know you might need to run for some more meetings, but if you can stick around, we have a couple more items on our agenda here.
Sure.
One is
I've got 2 more minutes. Yeah.
Okay. Stick around we're going to do the email bag, and then we'll do our science picks after that. Does that sound right, Vincent? Should we move forward? Okay.
Yeah, we have a couple of email this time, actually.
Oh, why don't we, since we got you for a couple minutes, actually, why don't we do our science picks, and then we'll circle back to the emails, because I think Nathan will probably give us the slip here in a few minutes.
Okay.
And Nathan, by the way, if you have, science picks, we're just this, if we've spotted something on the internet or an interesting, like if you have anything on your radar, I'll go first, we'll have Vincent go, and then if you want to add a guest pick, feel free to do so. So my science pick for this time, Vincent, is actually some of the local news coverage on Amanda, Maria, and Nathan's paper. There's one of the TV stations here, I guess the NBC affiliate, KSL, did a really great treatment of Nathan's paper here. And I'll let Nathan tell the story. That was my ulterior motive of going straight to the picks, because it's really great. So Nathan, why don't you walk us through that local news coverage here on your paper?
Great. Yeah, we thought that this might be of interest to people because people care about hair. So we worked with the university and put out a press release. And one of the people to contact us over it was Peter Rosen. And this man is a gem, absolute gem. He makes these features for news, and he's been doing, lifelong. Part of his origin stories, he's been working in media since he was 11 years old. Essentially, he was writing a weekly column back then. But anyways, he came and interviewed me in the lab about this paper, and then I thought, and he said it'll be on the local news. And then when it popped up there, we were just all completely blown away. He turned it into this kind of interactive poem with kind of animated naked mole rats running around, losing their hair. And it was just absolutely so much fun. So fun, poetic, and accurate at the same time. I'm just blown away by Peter and how well he did that. Yeah.
Yeah, we'll put the link into the chat, or we'll put the link up on Discord, or certainly at the TWiEVO website, but worth a look. It makes my, attempt at messing around with Joni Mitchell's lyrics sort of laughable compared to this professional treatment of the science here. And
it's cool.
And I kind of,
so I concede to this news report, but the word you said there, I think, is really important as well, Nathan, which is fun, and making our work, as we communicate it, or are trying to also, recruit other people to the profession as well, is to remember kind of how fun it is to explore genomes, to be genome hunters, and to go and then to kind of find these patterns that then can be very useful for biomedical reasons. But it doesn't have to necessarily lead to a cure for male pattern baldness or something like that. It's the pursuit of the science here, and how fun that is, and kind of centering and remembering that, which is why I really wanted to make this my pick of the week. Awesome. Vincent, how about you? What's your science pick?
So I have to say, when I did the polio genome sequence, we look at it and go, oh, look, there's a noncoding region at each end. Look, there's one long protein, and they're protease cleavage. I mean, all this stuff you can discover. We didn't have any programs at the time. We just looked at the sequence and we could figure stuff out. And so, yes, what Nels is saying, that you have a lot of sequences, you could do a lot more. But even one, the first time you see it, you find very cool things. We used to call it sequence gazing, right? Mm-hmm. It worked.
That's right. Yeah.
So my pick is a book by Ann Skalka. It is called Discovering Retroviruses. It came out a while ago, but I just did a retrovirus lecture yesterday in my course and was consulting it. And, it's a thinnish kind of book there, but it's the history of discovery of retroviruses, which, as you know, are very cool viruses. They have an RNA genome and they have reverse transcriptase that makes a DNA copy. It integrates and does all sorts of cool things. And so Ann Skalka has worked on them her whole career, and she's also a co-author on the virology textbook with us. So it's really good. A nice little book here for learning about retroviruses.
No, that's really cool. And, I think this is a case, at least for me, where it seems like there's one retrovirus that has taken up so much oxygen for the last several decades, which is, of course, HIV, being an ongoing pandemic. And so all of the diversity of retroviruses has sort of been pushed to the margins in some ways. And so, a book like this seems like to explore that genetic diversity. My God, the retroviruses are incredibly interesting, and I think that's kind of underrated.
I mean, the reason we picked up HIV so quickly is because they're immunosuppressive retroviruses that had been known in other species, in horse and in goats and cats and so forth, that did similar things. And so people said, hey, this looks familiar. So, and they were ready to make antivirals because they knew the targets. They had the reverse transcriptase, they had the protease, the integrase. So all this basic stuff on obscure viruses really, same thing with coronaviruses, right? It ends up being very useful in the end. Yep.
100% agree. Okay, Nathan, so we're putting you on the spot, but have you seen anything? It's fine too if you don't have a guest pick, no problem. But anything on your radar that you've seen in the last few weeks that have kind of science sighting out there that you wanted?
Yeah, well, I can bring up one here. There's this pretty neat paper we looked at in the lab a couple days ago. They were looking at Antarctic ice fish. So they're these really kind of cool fishes that have adapted to living in that extreme environment where apparently there's a lot of food down there. It's where the whales go to feed and all the plankton, and then they swim back up to Hawaii and don't eat for like 9 months or some crazy thing like that. But anyway, so these fish that live down there, they've been the study of all this kind of center of all this wonderful studies in the past, and how they deal with blood gases and things down there, and how they developed antifreeze proteins in their bloodstreams and things like that. It's just such cool work. Well, another study on them from Belinda Chang's lab in Canada, I want to say, I don't want to get this wrong, in Toronto, yes. They were looking at their vision, and it turns out a lot of these ice fish hang out right underneath the ice, right, where it's kind of, there's this blue light that permeates, and it's very different visually. And so they looked at kind of how the light processing proteins, the opsins and rhodopsins in our eyes, have changed within those species specifically to kind of adapt to that. And they showed there were kind of two main ways. One is that it looks like the wavelengths of light they've been newly tuned to pick up have kind of responded to that filtering that happens through the ice, which is really cool. And even a little more novel to me was the fact they also show this great evidence that it looks like the proteins have also just responded to just being cold, right? They had these changes that made them so they had, the kind of correct behaviors and things at basically 0 degrees Celsius, which is where they live all the time. And so that was pretty cool work. That was in molecular biology and evolution.
Very cool. And maybe another one of these natural experiments that you can look at, whether it's Antarctic ice fish or a retrovirus of a chicken. There's just so many, like all of this great diversity of biology unfolding right before our eyes. Love it.
Yeah,
Nathan, we
should know where such curiosity-based studies are going to take you, right? And as Vincent mentioned, it helped us in very practical ways, just kind of knowing what's around us, right?
Yeah, and couple that with
you have to let people be curious and you have to expect serendipity. And many people don't understand that. Why should we fund this? Who knows? Who knows what's going to come out of it? You have to take some risks, a lot of risks, I would say.
Couldn't agree more. That's sort of the motto of the show here. Until next time, stay curious. Well, Nathan, we should let you go. I know you have another meeting coming along here, but this is so fun. I'm so glad that you hopped on with us to take us on a tour of your paper. This is really great.
Yeah, thanks, Nathan.
It's such a pleasure, you guys, and for everyone for joining us. Thank you, and take care.
Bye-bye, Nathan.
Goodbye.
See you soon. All right, so why don't we open the mailbag? We've got a really nice letter here, Vincent.
Yeah, we do. Let me just move your name here.
Oh, yeah, there we go. We're back to our back to two co-hosts at the moment. But fun to host Nathan. I thought it was just like a fun coincidence where we got that recommendation on the paper. And then I thought, well, he's right across the hall. And I was pretty lucky to be able to time that out and get him here.
Okay. Yeah, we have one, because I guess I put one in that we had read. I didn't know that.
Oh, yeah.
So we have one. Why don't you take that one?
I will. So this is from Cian, writes, Dear Nels and Vincent, I'm a relatively new listener and have been going through the TWiEVO back catalog, and I wanted to get in touch and say thank you for this podcast. I'm currently taking a year out of studying before starting my degree in biological sciences in September, and this podcast has really fulfilled my desire for interesting and intellectually stimulating conversation in the meantime. I love learning about current research in the field, and it makes me so happy that there is so much going on at the interface between my favorite areas of evolutionary biology and genetics and genomics. TWiEVO is also a very helpful tool for me in the transition from learning by simply being taught the agreed upon science to learning instead about subjects that are currently under research and developing my ability to read, understand, and be critical of scientific papers. Any advice for someone about to start their degree in biological sciences who is especially interested in evolution? Many thanks, Cian, sent from the east of England, where it is currently a lovely 9 degrees centigrade. Well, listen, Cian, first of all, thank you for this massively kind note. I think both Vincent and I I'll let Vincent speak for himself, but we're both, I'm guessing, thrilled that the back catalog, the 86, now 87 episodes have brought you some positive scientific energy. And, you've put your thumb on exactly what we're trying to do, is to really pull out the kind of the concepts that are at the kind of cutting edge of the field. And, that was what we hopefully were able to do today with having Nathan on the show as a guest as well. The interfaces between computational biology, genome scale analysis within experiments. And if you don't actually lift up a pipette, but you take that experimental view or think of sort of the natural examples of experiments and how that, I think, can kind of sort of push the work or the insights to the next level. I think the Clark Lab is really sort of on the masters of this. So for me, this is also just so I feel like I'm still learning, transitioning in my own career. And so that's exactly, I think the area to point to. Well, what do you think, Vincent? Any advice on someone about to start their degree who's interested in evolution?
Just try and get as broad an education as you can in biology, right? You need to know all the underpinnings. You need to know some biological chemistry, right? I mean, Nathan did organic chemistry for a while, but I think that's really important, so you understand how things interact. And you have to understand other aspects of biology, molecular biology, genetics, even, of course, you want to take some evolution courses, but make sure you go beyond that. Don't just take evolution, get everything under there, because that's, as you heard today, you need to know the biology to understand selection forces in evolution. Otherwise, you can't do that. So, go to a place that has a good mixture of all of that. Make sure you look at it and see the courses they offer, and hopefully you'll find a place where there are people working on some of these things, not just a teaching school, but a research school, and that would be great because then you can go talk to people and maybe do some research as well. That's what I would say.
That's great advice. And I think that's, what that opens up is the possibility of taking, whether it's organic chemistry or another field and sort of bringing it to the field of evolutionary biology. That's sort of how we, as we're picking papers or topics or just, kind of pointing our discussion in one direction, that's what we're really looking for, is someone kind of coming at this by taking a technique or a method or a viewpoint or a concept and bringing it to evolution, maybe in a new way. I guess I would add to this one other small point, which is also, sort of kind of browse like widely, be an omnivore of science, as Vincent, I think you're describing. And then at the same time, but also listen to your own gut or your own heart in a sense. And so, there might be like the perfect topic that's just about to blow up the field and you can kind of see it all, but it might just be that you like read about it and it's just boring. So there might be entire topics that like would make sense scientifically or from sort of a business standpoint of becoming a scientist or continuing science in some way. But there's something about your own curiosity or your interest that you're also, I think, feeding and nurturing in this process of as you start a degree in biological sciences. And so I also recommend paying attention to that, is just follow your nose. Like I'm guessing for Vincent and I, we wouldn't have guessed maybe at this career step that would be the viruses that would just capture our hearts and like wouldn't let go in some way. For other people, it's bacteria or naked mole rats or whatever the system is. And so it's okay, I think, to also explore that and just things that you just can't help but be fascinated by and let that sort of build and grow as well.
Yeah, you're gonna have things that grab you, and then you have to find someone who can further it. Right. So I got grabbed by viruses, and then I found someone whose lab would help me further that. If you can't do that, then it's not going to work. So the two are really important. But you have to find something that gets you curious about it from the start, and that's going to start the whole process going.
Yeah, agree. No, thank you, Sian. Great letter, and good luck as you keep us posted on that journey as you transition back into school.
Yeah, please do. Well, if you wanted to send us a letter, I know many of you come here and ask your questions here, but if you want to send us a letter, you can send it to TWiEVO at microbe.tv. And microbe.tv/TWiEVO is where you can find all of the show notes and so forth, links to these episodes. And if you enjoy what we're doing, we'd love to have your financial support. You can go over to microbe.tv/contribute. Nels LD is at cellvolution.org. On Twitter, he's L Earlybird. Thanks, Nels.
Hey, thank you, Vincent. Great to be with you again. Fun to have a guest, and keep healing up. Drink some, tea with honey or something to take care of your voice. That's an important asset.
I think it is. And thank you for getting Nathan. Nels told me last night, I think, by text. Yeah, that's how we communicate at all hours. Right. So I'm going to get Nathan on this. That's great. Wonderful. Because it's always nice to hear from our colleagues, right? You hear from Nels and us all the time, but our colleagues are interesting. And a guy like Nathan obviously likes to talk. So it's a good person to hear right from the researcher's mouth what's going on in their head.
Yeah, agree.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. I want to thank everyone for coming today. We had 100 people here at some point, so that's a new high. Thanks so much for that. I want to thank our moderators. Andrew is newly joined moderator.
Fantastic.
Barb Mack, Les, Steph, and Tom were all here today. Thank you so much for moderating. This is a little bit of a quieter audience than the live streams, so there's less moderating work, which is good for you. So anyway, thanks for doing that. And here we have a contribution from Rob. Thank you so much for your contribution to science education, and thank you, Nels, for doing it again. Really appreciate it.
Appreciate it here too. Thanks, everyone. Looking forward to the next time already.
You've been listening to This Week in Evolution. Let's get the music going, Nels. How about that? Podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, Stay curious. Bye-bye.